Hello, my name is Tatsumi Hirata. I work at the National Institute of Genetics in Mishima, Japan. This is Dr. Ikuo Suzuki, leading author of a paper published in Developmental Cell. The higher brain function in mammals is thought to come from their highly evolved layered neocortex. It's called the neocortex because it's assumed to be a newly evolved structure in mammals. Ikuo will explain that this is not new, but is in fact constructed by an ancient mechanism. Please listen to his findings.
In all mammalian species, the neurons in the neocortex are horizontally organized into deep and upper layers. This is unique to mammals. The first step of this study was finding that although the non-mammalian chick doesn't have layers, it does have neurons that are molecularly and functionally homologous to the deep and upper layer neurons of the mammalian neocortex. But rather than forming layers, they are clustered into two separate domains within the region homologous to the neocortex called the parietum. We found the reason for this separation. In the chick, the deep and upper layer neurons are produced from separate brain regions. Specifically, the stem cells in the medial region produce only the deep layer neuron, and the stem cell in the lateral region produce only upper layer neuron. This is very different from mammalian neurogenesis. In mammals, all the stem cells, regardless of their position, produce both deep and upper layer neurons. The deep layer neurons first, and then the upper layer neuron. This mechanism creates horizontal neuronal layers across the entire region. However, much to our surprise, even a chick possesses the same potential. It's just hidden in vivo. When the chick neural stem cells isolated from the parietum and cultured in vitro, they can, in fact, produce both deep and upper layer neurons in the same order. Both the medial and lateral stem cell have the same potential. We explain the result like this. The chick stem cells intrinsically have neurogenetic potential very similar to that of mammalian stem cells. However, this potential is normally suppressed by local factors in the environment of the chick brain. As a result, in vivo, the chick stem cell produces only one of the neuron subtypes.
When we started this project, I was thinking that the mammalian type neurogenesis was specific to mammals and co-evolved with the layered neocortex. So it was quite surprising that the chick, which doesn't have the layered neocortex, intrinsically has the mammalian type neurogenetic potential.
The important message of this study is that the last common ancestor of mammals and birds probably already had this potential, and the two neocortical neuron subtypes. The potential is inherited by mammals, birds, and even the extinct dinosaurs. Each animal group may have modified the neurogenetic process in a different way during evolution, thereby creating their own types of neuronal arrangement.
So it's kind of ironic that in the chick, the regulation of neurogenesis by environmental factors is more complex than the simple mammalian system. In a way, mammals have gotten rid of spatial restriction and taken full advantage of the intrinsic multipotency.
Yeah, that's right. As you say, the mammalian scheme is much simpler than the chicks, but we still don't know about the neuronal arrangement in the common ancestor and what happened during the split of mammals and birds from that common ancestor. To answer this question, we need to study the other amphibians and reptiles living today, such as frogs, lizards, and turtles. That will be the next step in our project.